In this presentation, I will discuss the values and best practices we've been following in developing an integrated application for skin safety assessment. The SaferWorldbyDesign initiative aims to develop a platform providing integrated testing and assessment, supporting the design of safer products. For such a goal, shared values and best practices are key aspects of our vision for building a reliable platform, supporting integration of evidence and collaboration. SaferSkin was developed initially as a web application to predict skin sensitization, supporting the work of risk assessment of product ingredients. We expanded the scope and vision of SaferSkin within SaferWorldbyDesign as a collaborative application supporting the integration of data and models from different partners, including in vitro testing of ingredients and formulations, and extending the endpoints assessed. Data integrity is an important value we pursue in our solution development. Lack of data integrity can lead to significant issues. For example, as reported in June 2020 in Science, two elite medical journals, The Lancet and the New England Journal of Medicine, retracted coronavirus papers over data integrity questions. Data integrity is the maintenance of and assurance of data accuracy and consistency over its entire life cycle, including experimental design, trusted data management, application implementation, and reliable user consumption of the data for a specific purpose. We aim to support data integrity goals from its initial generation through to its downstream consumption. Increasingly, we have been building consensus on approaches to data sharing within the community. As for the examples shown here, between European research projects in nanosafety and associated interactions with other international programs such as in the United States and Asia, currently progressed under the NanoCommons Community Infrastructure Initiative. These efforts should help enable a sustainable and harmonized knowledge infrastructure supporting the risk assessment of nanotechnology. This brings us to a core question. How can we ensure data integrity in communications of scientific evidence? Although legal and confidentiality issues bring significant challenges, there is much we can do to improve the quality and integrity of data associated with scientific communications and use of data. Examples include the specification and adherence to processes that map out our data formats and transformations, use of technical and procedural controls to prevent data, metadata, or settings to be deleted, amended, or excluded without authorization, restriction on deletions or modifications, documenting changes, producing new versions of datasets, curation of data, timestamping, preserving true copies of complete data, including raw and processed data, documenting processing protocols, recording of accesses and changes, maintaining audit logs, and use of validation steps to verify data and data mappings. The SaferSkin application supports the integrated testing and assessment of an ingredient against the adverse outcome pathway for skin sensitization, including the molecular initiating event with skin proteins and the downstream immune system response. 3 OECD-approved assays may be used to test a compound against the key events of the adverse outcome pathway. Defined approaches use different algorithms applied to the data to determine a classification, for example, non-sensitizer or moderate sensitizer, and when possible, to predict a potency, that is, a lowest effect level that can be used in risk assessment. A multivariate regression approach can be used for developing a predictive model for the endpoint, both including global and local model approaches. Our Bayesian network approach builds a weight of evidence across in silico descriptors and in vitro assay data to determine a prediction with an uncertainty and a confidence. The method can be extended to include additional data from newer assays for building new models. The SaferSkin workflow starts with the entry of a chemical structure. SaferSkin calculates physical-chemical descriptors based on the chemical structure. Experimental values from the user can also be used to override default values. The user can enter experimental values for assay data into the user interface. SaferSkin prediction results for the endpoint are generated for different defined approaches, which can be compared. SaferSkin finally generates a report with the details of the calculations and the results. We have developed and share here a tutorial where the user can work through the different steps in preparing and sharing data. We use PCR as our example, as one of the most common biological experiments in the world, both in research and testing. To start with, we need to work through the preparation and processing of raw data. Metadata needs to be prepared to describe the dataset and the data organized into a format labeled by the metadata. The data can then be processed by software into a database. This procedure can also be elaborated through the use of validation software to check the validity and completeness of future datasets. Once the data has been uploaded and validated, we can use the metadata and data to provide access to the data through well-defined instructions and information provided through application programming interfaces, which can be used as services to support many different kinds of tasks and web applications. We provide an example of using notebook technology linked to the database interfaces to describe our procedure in a web-based format, including interactive analysis and visualization of the data. The code supports the execution of the data analysis, which can be viewed on the web browser and also followed programmatically. Because metadata has been captured with the dataset, it can also be provided to the documentation of the results. Changes can be made in the code and the analysis updated interactively. The code documents the procedure. An example is shown here for preparing the metadata for a skin irritation assay following the above tutorial example. The raw data, as generated on the screening plate, is provided as part of the analysis. A comparison of the results for different products and replicates is generated and provided to the user through a web-based interface using data from the SaferSkin cloud. Reproducibility of results is an important goal we pursue, as discussed at the Global Regulatory Science Conference in 2017, and including documentation of both in silico and experimental protocols and processing of data. Trust is an important goal we are aiming for on data and documents used in the supporting knowledge infrastructure. We are pursuing the goal of trust in the supporting infrastructure and including consultation with the community on best practices, qualification, and acceptance. In summary, SaferSkin has shown us that working on developing best practices within the context of scientific application development is a worthwhile endeavor, and one which we can work on productively, both individually and in collaboration.